Let's begin with the first category of actives, the postbiotics. The postbiotics are a platform of specialties for microbiome care with multiple references, each of them specific for diversified applications and with a diversified composition. We have Kalibiome Age and Kalibiome Sensitive that are targeted respectively for skin anti-aging and treatments for sensitive skin, and that will be the object of today's webinar. Just a quick mention of the other two actives included in the platform that will not be discussed today. One of them is Kalibiome Ecogenetic, which is specific for the daily microbiome care. The other one is Kalibiome Auris, specific for oral care treatments. Kalibiome Postbiotics are a new call of active specialties that represent the future of cosmetics industry. Today, the maintenance and strengthening of skin microbiota is the key to guarantee the wellness and a good functionality of the skin. Kalichem is introducing a new class of specialties for dermatological applications that represent the most advanced frontier of microbiota care. This innovation was developed by the laboratories of the Humanitas University of Milan, with the scientific team worldwide recognized as one of the most relevant players in the knowledge of microbiology and microbiota. Now, let's see in detail what this innovation consists of. In compositional terms, Kalibiome postbiotics are oligopeptides, fatty acids, and biosurfactants released by Lactobacillus paracasei through a controlled biofermentation, enabling to obtain the above substances in consistent amounts and with a unique purity yield on a reproducible basis. In functional terms, these postbiotics are the messengers and the actual effectors of the bacteria beneficial actions on the skin. Contrarily to probiotics and similar actives, Kalibiome postbiotics show a unique safety profile because of the total lack of bacterial fragments, bacterial wall, and toxins in their nature. This optimized safety profile is demonstrated in a scientific publication of a medical journal, Gut, through a study carried out on the main headquarters of the bacteria in the human body, the gut indeed. This study proves that individuals affected by the Crohn's disease, a pathology characterized by prolonged events of local acute inflammation, when treated with commensal bacteria of the Lactobacillus family, show worsening of inflammations, whereas the treatment with postbiotics obtained through the Kalibiome technology shows maximum efficacy, tolerability, and a total lack of side effects. From a technological standpoint, Kalibiome postbiotics are obtained through a patented biofermentation process, implying 2 steps of fermentation rather than 1, with the second step carried out in a minimal culture medium through specific conditions which vary depending on the postbiotics output required. The knowledge of bacteria behavior and response during fermentation, along with the knowledge of the postbiotics' nature and selectivity of action, allows to design diversified groups of postbiotics with diversified activities. Kalibiome technology was awarded at the IFSCC 2019, an extremely prestigious accomplishment, keeping in mind the strict parameters imposed by the judging scientific board of this organization. Following a table with an overview of the main advantages that Kalibiome postbiotics bring to the table compared to other microcategories of actives for microbiome care. In comparison to the prebiotics, which are the food of bacteria, Kalibiome postbiotics show higher specificity of action given by the presence of selected actives with specific function in their composition. Prebiotics instead are food for bacteria with no specificity or specific targeting. In comparison to the probiotics, which are living bacteria, we have 3 different types of added values. First, technological advantages, since Kalibiome postbiotics do not contain living cells. As a result of this difference, there is no theoretical or practical possibility of contamination or induced bacteria inactivation triggered by cosmetics preservatives. Another consistent technological benefit is linked to the storage condition. Given their chemical nature, Kalibiome postbiotics do not require refrigeration. In the second place, we have safety advantages since Kalibiome Postbiotics do not contain living bacteria, bacterial fragments, bacterial wall, or toxins. In the wake of the previously mentioned study, Kalibiome Postbiotics show maximum safety and tolerability even in presence of acute inflammations. The probiotics instead, even commensal ones, in acute inflammation scenarios, could worsen the tissue's damages. The third advantage is functional. The probiotics, in order to give a beneficial action, need to crosstalk properly with the skin. Such crosstalking is quite facilitated in individuals with normal conditions, but it gets highly critical in individuals with uneducated skin. Now, one of the main examples of uneducated skin corresponds to the atopic skin. Individuals with atopic skin have unpredictable reactions when they get in contact with bacteria, even commensal ones. Kalibiome Postbiotics, given their total lack of bacteria and their related structural components, overcome the need of crosstalking with the skin as they are indeed ready to use actives whose benefits are observed regardless of the education of the skin. Other actives used in the market are the paraprobiotics, which are heat-inactivated bacteria or tyndallized bacteria. Their main benefit in comparison to the probiotics is linked to their higher stability in formula. When comparing them to Kalibiome Postbiotics, they still show the same criticalities of the probiotics in relationship to the cross-talking mechanisms with atopic skin and uneducated skin, as well as safety matters in individuals with acute inflammations and uneducated skin. The last category are the lysates and ferment filtrates. These products are obtained through a standard traditional fermentation carried out with a single step where the bacterial output can be filtered or not. Compared to these actives, Kalibiome Postbiotics show the following advantages. First, a higher content of reproducible and specific actives given the different criteria of the fermentation conditions. Second, a higher efficiency of action given the postbiotics' low percentage of use required in formula that starts from 0.1%. And three, optimized organoleptic features since Kalibiome Postbiotics come in lyophilized powder form. Here summarized the focal points of the technology. Kalibiome specialties are made of concentrated and reproducible actives obtained through a patented biofermentation. Their composition is totally bacteria-free and only containing active ingredients like peptides, fatty acids, and biosurfactants. They are preservatives-free, they can be stored at room temperature, they have no interference with cosmetics preservatives, and they show high efficiency of action given by the low dosages required, which are between 0.1 and 0.5%. Now, an overview of the first active ingredient of the family, Kalibiome Age, specific for advanced anti-aging treatments. As a first step, let's take a look to which are the targets of the Kalibiome Age. First of all, let's focus the attention on the epidermal target because this active ingredient proved efficacy in restoring the basal epidermal hyaluronic acid. We know that this substance at this level is involved in activities like cell differentiation regulation, skin hydration, organization of the framework of the skin, and skin healing. At dermal level, Kalibiome Age targets the collagen 1 network regeneration and the repairing process of fibroblasts. This is quite important as an effect for the anti-wrinkle activity because we know that a solid collagen structure is essential to promote skin resilience against the deformation and daily stress. The activities on hyaluronic acid, collagen, and fibroblasts are related to a selective targeting of the postbiotics towards specific cells receptors. Among the targets, we have EGF and toll-like receptors, receptors coupled to G protein, whose activations trigger cells proliferation, migration, and control of the inflammation leading to anti-aging. In vitro ELISA tests demonstrate that both the basal hyaluronic acid produced by keratinocytes and the dermal collagen produced by fibroblasts are increased more efficiently and effectively by Kalibiome H when compared to a competing bacterial ferment found in the market. In detail, 10-fold lower concentration of Kalibiome show better performances than the benchmark. A scratch test demonstrates Kalibiome H superior activity when compared to a competitor containing bacterial ferments and an overall pattern matching the performances of the EGF. On the right side, we can observe the fibroblast repairing pattern after 24 hours. Following a summary of the 6-week in vivo test carried out on a Kalibiome-based cream with evaluations of skin replenishment and redensification, anti-wrinkle action, and finally skin firmness and tensor effect. For each of the above studies, we have analyzed through metagenomic assessments the variations of microbiome by analyzing which bacteria increased or decreased. Just a quick view of the metagenomic tests. These are detailed studies enabling to measure through the ribosomal RNA the variations of the microbiome following the treatments with the active and the placebo. The skin redensification test shows through a digital image the increase of the collagen content inside the skin following the treatment with Kalibiome. The images show that after a 6-week treatment, the white bands corresponding to the dermal collagen are significantly increased compared to the value at time 0, with a consistent increase of the skin density of approximately plus 38%. In quantitative terms, the increase of the collagen content in the dermis is expressed through the value of the skin echogenicity. The results prove an activity of Kalibiome 12 folds higher than the placebo cream. From a cosmetics point of view, this activity can be exploited to develop cosmetics counteracting the photoaging process, hence promoting the skin resilience against UV rays exposure. The subjects that reported increased skin redensification after the treatment with Kalibiome H showed significant variation of the bacteria Prevotella and Faecalibacterium. In detail, there is a positive correlation between the increase of the skin redensification and the increase of the concentration of the bacteria Prevotella nigricans, and a negative correlation with the bacteria Faecalibacterium. In other words, very simply, this means that when the skin redensification increases, the Prevotella bacteria tends to increase, while the Faecalibacterium tends to decrease, as clearly reported in the 2 graphics on the left side of the screen. Following the first of 2 examples of how the treatment with Kalibiome H impacts the wrinkle appearance. As a result of the skin redensification and the increased skin collagen content seen before, the skin replica images highlight the reduction of both wrinkle width and depth. And here, the second example where the activity pattern is the same as the one seen right before. The following graphics show in quantitative terms the combined reduction of the wrinkle width and depth according to the RA and RZ parameters, still by comparing the activity of Kalibiome H and placebo. In relationship to the microbiome variations analyzed, the tests highlighted a correlation between wrinkle reduction and increased expression of the bacteria Stenotrophomonas and Brevundimonas diminuta. In detail, wrinkle depth reduction is correlated to increases of the expression of the Stenotrophomonas, while wrinkle width reduction is correlated to increases of the Brevundimonas diminuta. The final in vivo test shows the efficacy of Kalibiome H in reducing skin extensibility by 76% when compared to the placebo treatment. Decreased extensibility is a hint of increased skin firmness and tensor effect, according to the evaluation of the R0 parameter of the cutometer. Getting back to our microbiome metagenomic assessments, we can observe a couple of positive correlations between increased skin firmness and tensor effect values and the increased concentration of a couple of bacteria, in this case the Alister and Alloprevotella tannerae. For the skin firmness parameter, we also have some negative correlation because the subjects that reported the best results in terms of skin firmness increase reported reductions of Crysiobacterium and Paracoccus bacteria. Following a quick recap of all the benefits linked to Kalibiome age. This reference is a candidate active for N-Cosmetics claiming hyaluronic acid and collagen increase, skin redensification, anti-wrinkle activity, elasticity and skin firmness increase, resilience against photoaging, and a refined microbiome regulation as demonstrated by the metagenomic test that we have shown previously. The second reference of Kalibiome Postbiotics family is a specialty specific for soothing and protecting sensitive and atopic skin. Trade name Kalibiome Sensitive. One of the main functional targets of Kalibiome Sensitive on the epidermis is the zonula occludens 1, also known as ZO1 tight junction, which is a protein able to seal and keep compact between each other the keratinocytes, in order to promote and maintain the skin barrier integrity, form an anti-pollution shield counteracting dehydration and protecting the skin against external aggressive factors, including pathogens. The following in vitro assessment shows through fluorescence skin keratinocyte cells, highlighted in blue, subject to a previous treatment with SLS patch and following treated with a control, as in the upper image, and Kalibiome sensitive in the lower images of the graphic. The ZO1 tight junction is highlighted in green through immunofluorescence, and it is visibly much more expressed in the lower images corresponding to Kalibiome sensitive treatment. Their high expression enables to keep the keratinocytes strongly linked between each other, as clearly visible from the picture, therefore increasing the integrity of the barrier that they are able to form. In order to quantitatively assess the integrity of the keratinocyte barrier, a test of transepithelial resistance was carried out. In a system containing two compartments separated between each other by a microporous membrane laying on a keratinocyte cell monolayer, an electrode connected to a voltmeter was used to evaluate the barrier integrity according to the electrical resistance generated. If the cell's junctions are strong, the values of resistance measured by the electrode are expected to be high, otherwise they are expected to be lower. The in vitro test shows that cells infected by skin pathogens and following treated with Kalibiome sensitive show a barrier integrity absolutely comparable to that of healthy and non-infected skin cells, whereas in all the other cases of infection and lack of Kalibiome treatment, the values are significantly lower. This test is an evidence that Kalibiome sensitive is able to protect the skin barrier integrity even in presence of pathogens on the skin. The following tests show the effects of Kalibiome sensitive on atopic skin in relationship to the soothing effect against itching and hypersensitivity related to atopic dermatitis, and how Kalibiome counteracts the formation of the biofilm formation of the Staphylococcus aureus. The following skin biopsy images show on the left side the lesional skin typically occurring in atopic dermatitis, and on the right side the healthy skin. We can observe the different expression of a specific immune system protein, the long TSLP, highlighted through red fluorescence. This protein, the thymic stromal lymphopoietin, in atopic dermatitis in its long isoform is occurring 8 times more than in healthy skin, and it represents a mediator of itching and inflammation, the typical effects of atopic skin. Kalibiome sensitive was proven to decrease such itching protein to its homeostatic values and increase its physiological anti-inflammatory isoform, the short TSLP. ELISA assessments prove that Kalibiome, when compared to control and poly-IC, is able to decrease the itching TSLP isoform by 8 folds and to increase by 3 folds the short anti-inflammatory isoform. Let's take a look now to an interesting and interactive test carried out directly on plates containing the Staphylococcus aureus bacteria. Such bacteria was cultured in a plate and following treated with a control and Kalibiome sensitive. Let's see what happens. The cyclic videos you're about to see show in both the plates some black dots corresponding to the bacteria. Now, the more the black dots grow in number, the more the bacteria will tend to form a biofilm. That's what happens in the case of the control especially. In the case of Kalibiome, instead, you will see particular moments where the bacterial expression will be totally inhibited. That's the activity of Kalibiome-sensitive biosurfactants that will physically inhibit the bacteria quorum sensing, therefore inhibiting their communication between each other, and exert a final bacteriostatic action. For time reason, we will not detail other in vitro tests, but just for your information, it is worth mentioning the activity of these Kalibiome postbiotics also in modulating the expression of several cytokines involved in the reduction of the inflammation process. For example, there is activity on interleukin 10, 6, 8, 12, and others. Among the most relevant in vivo tests carried out, we have reported increases of skin hydration of plus 20% after a treatment of 6 weeks. According to the metagenomic tests, the hydration increase is correlated to increased variations in the microbiome of the bacteria Rhodococcus, which is correlated to the increase of the skin moisturization. Additional in vivo tests prove an overall reduction of transepidermal water loss within 72 hours of minus 40%, with a plus 13% score reported against the placebo treatment. In such a case, the skin barrier integrity improvement was correlated to a variation in the microbiome of the Brevundimonas diminuta, still accordingly to our metagenomic tests carried out. Finally, we have an in vivo evaluation of the skin redness reduction calculated through the chromometer by measuring the Hunter coordinates, more specifically the A value coordinate with a percentage reduction reported of minus 15%. Now a quick recap of all the features seen so far. So Kalibiome sensitive is a candidate for N-cosmetics claiming an itching and reddening reduction activity, soothing atopic and sensitive skin, promoting the integrity of skin barrier, reducing inflammation, preventing dark circles, anti-pollution activity, and much more.